This is how lactate alkalinizes your muscles. You heard that right. Not acidifies, alkalinizes your muscles. I'm Dr. Chris Masterjohn. I have a PhD in nutritional sciences, and you are watching a masterclass with Masterjohn energy metabolism lesson. This is a course where we break down the biochemistry of how we use the energy in food to fuel all the important processes in our body that contribute to our wellness, performance, and longevity. And in this lesson, we're going to look at the biochemistry of how lactate is alkalinizing. You may have heard that lactate is something that is produced when our muscles make lactic acid when we're exercising, that acidifies our muscles and contributes to fatigue, failure, and delayed onset muscle soreness. Or you may have heard the arguments against this. There are many well-known people who have been spending a long time, big parts of their career, countering this narrative, like Andy Galpin. You may have heard, if you're deep in the science, you may have read the work of George Brooks. We're going to take a deep look into the biochemistry. We're going to learn how we do not make lactic acid in our metabolism. We make lactate. When we make lactate, it is alkalinizing. We're going to look at how some of the presentations in biochemistry textbooks, on the one hand, give us everything that we need to understand why it is lactate that we make and not lactic acid, that we never make lactic acid in our metabolism. But on the other hand, do not equip us very well to understand what actually is the source of acidity in exercising skeletal muscle or in metabolism in general, because they don't put enough emphasis on acid-base balance to bother showing you with completeness where all the protons, which are the cause of acidity, are going in the chemical reactions. So we're going to take a look at the Berg and Lehninger presentations of glycolysis to see where they go wrong in equipping us with the knowledge we need to understand acid-base balance during exercise. And finally, we're going to end with some practical implications. We'll talk about what actually does cause fatigue in exercise skeletal muscle and exercising skeletal muscle. And we will also look at the research on lactate supplements briefly to see how we can leverage what we learn in this biochemistry towards how we should interpret the human trials with lactate supplements in the context of other ways that we could leverage nutrition and supplementation towards improving exercise performance. So super briefly on the screen is the typical presentation of glycolysis divided into two phases based on investing energy versus generating energy, where we invest 2 ATP, we get as a payoff from that, it's also called the preparatory and payoff phases. As a payoff, we get 4 ATP, so that's net 2 ATP gain, and we also get 2 NADH. We're going to now look at it from a different perspective, how we could divide it between acidifying phases and alkalinizing phases. Before we do that, just to make sure we're all on the same page, I want you to think of a proton as a hydrogen ion, as an H+, and as acidity. These are all the same things. The simplest element is hydrogen, 1 proton, 1 electron. Take away that electron, you form the positively charged hydrogen ion, which is the same thing as a proton. The accumulation of protons is the cause of acidity. We can measure it and express it as the pH. The P in pH means the negative logarithm of the hydrogen ion concentration. Because it's negative, the lower the pH, the more acidic. The higher the pH, the less acidic. Because it's logarithmic, as we go 1 unit on the pH scale, we're moving by a factor of 10. So 7 is neutral, 6 is 10 times more acidic, 5 is 100 times more acidic, 4 is 1,000 times more acidic, and so on. If we divide glycolysis into the acidifying and alkalinizing phases, we are going down to the production of 2-phosphoenol pyruvate. So we're going almost to the end of glycolysis as the acidifying part. And the alkalinizing phase is the pyruvate kinase reaction and the lactate dehydrogenase reaction. I'm depicting lactate as the end product of glycolysis because there's a rich body of evidence that, contrary to the presentation in the textbooks, lactate is the dominant, if not the overwhelmingly or nearly exclusive end product of glycolysis. The protons released are fractional because it's not only the case that we have covalent bonds changing as we change the construction of a molecule that may release a stoichiometric amount of protons or consume them, meaning we can mathematically account that as we go from one lactate to one pyruvate, we must do one or another thing with the reactants and substrates involved that add up into a mathematical formula where everything is expressed in whole numbers. In addition to that, we also have hydrogen ions that are binding to or releasing from the substrates and products in these reactions, and we can express those as probabilities. So we have an average fractional proton yield. We will talk about that, but to simplify things here, I've rounded these all off to whole numbers. And when we do that, to make it easier to understand, we are releasing 5 or 6 protons in the acidifying phase of glycolysis, 5 if we come from glycogen, 6 if we come from glucose. And then during the alkalinizing phase, when we end at lactate, we're consuming 4 protons. That is net acidic regardless of whether we came from glycogen or free glucose. But if we remove lactate from the cell, we have to remove 1 proton per lactate molecule because of the way that the transporter is designed. And that means that if we break glucose into 2 lactate and remove 2 lactate from the cell, we remove 2 more protons. That is a net clearance of 6 protons in the alkalinizing phase, and that will allow us to break even if we came from glucose or to be net alkalinizing if we came from glycogen. As brief review, these are the 10 reactions of glycolysis. We phosphorylate glucose to glucose 6-phosphate, not shown here. We also could have broken glycogen down to glucose 1-phosphate and isomerized that to glucose 6-phosphate. Then we isomerize the glucose 6-phosphate to fructose 6-phosphate. We then phosphorylate that to fructose 1,6-bisphosphate. That is with the enzyme phosphofructokinase, or PFK. Super important enzyme because it's the first committed step in glycolysis. You could have done other things with this glucose 6-phosphate, and also because it's highly regulated. So with respect to our discussion, the most important regulation of it is that it is strongly inhibited by acidity. The fructose 1,6-bisphosphate then gets split apart into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. If glycolysis is proceeding smoothly forward, the dihydroxyacetone phosphate converts to a second molecule of glyceraldehyde 3-phosphate. This concludes the energy investment or preparatory phase. It now begins the energy generating or payoff phase, and it also marks the point where we have 2 of everything now because we've split the molecule in half. So everything here happened once, everything forward is going to happen twice. So 2 molecules of glyceraldehyde 3-phosphate are going to get oxidized by 2 NAD+, generating 2 NADH and 2 protons, and that's going to form 1,3-bisphosphoglycerate. In doing that, we are using the enzyme glyceraldehyde 3-phosphate dehydrogenase, or GAPDH, and that is going to be another super important enzyme because it's an NAD+ sensitive step, the one NAD+ sensitive step of glycolysis. So we're going to need this NADH to be in net oxidized in the respiratory chain in order to allow this step to go forward. And it's also the most acidifying step in glycolysis. We're going to see additional reasons why that's true momentarily. Then 1,3-bisphosphoglycerate, we have 2 of those. That produces 2 ATP, yields 2 molecules of 3-phosphoglycerate. That gets isomerized to 2 molecules of 2-phosphoglycerate. That gets rearranged using water to 2 molecules of phosphoenol pyruvate. That then produces 2 more ATP for a net total production of 4 ATP and a net production of 2 ATP, yielding 2 molecules of pyruvate. This then stops, as is typical at pyruvate, only as I said before, we are always or almost always going to actually continue going on to lactate. But that concludes the typical presentation in a textbook and in most online sources of the 10 steps of glycolysis. All right, so here is the redox/acid-base reaction that occurs with NAD plus and NADH. NAD plus, in the upper left, we can see that it binds 2 electrons and 2 hydrogen ions. One of those hydrogen ions becomes NADH, and the other is left in solution. One of those electrons was neutralizing the positive charge on the NAD plus. The other was gluing that first proton to the NADH molecule. That can also be written as NAD plus accepting a hydride ion, which is an H negative instead of an H plus. That's a proton with 2 electrons. And that becomes NADH, and there's 1 proton that's left over. Either way, because we have 2 electrons with electric neutrality, we have to balance that with 2 protons. One of those protons is always going to be released into solution, simply to account for the fact that NAD plus has 1 electron neutralizing its positive charge and 1 getting added on as NADH. So when you hydrolyze ATP, as hydrolyze implies, you're using water. And the reason that you're using water is because to break apart the terminal phosphate, you have an oxygen that is essentially shared with the phosphate next to it. And you're going to have to reconstitute the oxygen of the phosphate that you're ripping off if it can no longer share the oxygen with the phosphate next to it. So you take the oxygen from water to do that, and one of the hydrogens of the water will tend to stay on the phosphate molecule, but the other will be left into solution as an extra proton. Conversely, if you synthesize ATP in the mitochondrial respiratory chain, you go the opposite way. You do dehydration synthesis, and you remove the OH from the phosphate with a proton that you've taken from solution, and that will reconstitute the ATP molecule, and you will have removed a proton from solution. So ATP synthesis is alkalinizing, and ATP hydrolysis is acidifying. However, the substrate level phosphorylations in glycolysis do not use dehydration synthesis. And you can see in the Lehninger textbook that depicted why in the case of the phosphoglycerate kinase reaction. They've shadowed in this pinkish orange color a box around what is moving, and it's not actually phosphate, it's phosphite. It's PO3, not PO4. And you do not need to reconstitute it. You do not need to reconstitute its 4th oxygen because you're not generating free phosphate in water. Instead, you're taking PO3 and you're leaving behind the oxygen of the substrate, and in this case, you're allowing it to become a carboxyl group, and you are moving PO3 onto the adenosine diphosphate molecule. Now remember, adenosine diphosphate, that phosphate on the end, it has its own oxygen. So you don't need to take out OH when you move it there because you're just taking PO3, you're not taking PO4, and you're just putting PO3 where PO3 needs to be. You're not dealing with any extra oxygen. So in the substrate level phosphorylations of glycolysis, there's no 4th oxygen that ever needs to be dealt with. There's no hydrolysis. There's no dehydration synthesis. There's just a transfer of a PO3, and there's no proton exchange. However, in the second substrate phosphorylation reaction, you do happen to absorb a proton for a completely different reason. So here you have the same type of transfer of PO3 from phosphoenolpyruvate to ATP, but this hydrogen ion that you see getting used is not doing anything with the phosphate. It's actually making the last hydrogen on the carbon show up. The way this happens is that as the PO3 leaves, the hydrogen ion from solution comes in and gets added as an OH. This is what's called an enol form of pyruvate. And the enol is a double bonded carbon with an OH coming off of it. Enols tautomerize to ketones. Ketones are a double bonded oxygen in the middle of a molecule. In general, whenever you have an enol like this, it's going to tautomerize to the ketone form. And what we typically think of as pyruvate is the ketone form. And so that hydrogen that came in on the oxygen winds up down at the bottom on the carbon. So that's the reason that a proton is being used up in the pyruvate kinase reaction. Phosphorylations and dephosphorylations of sugars within glycolysis do release protons. Although this is not a case of hydrolysis and dehydration synthesis, this is a case of exchanging the proton on the end for the PO3 or phosphite that's going to come off of the ATP molecule. So PO3 jumps off the molecule, hydrogen jumps off the molecule as a proton, and the PO3 gets put in the proton's place. And so in each phosphorylation reaction at the beginning of glycolysis, you do release one proton. All of this becomes quite a bit more complicated when we think of the fact that both phosphate species, as well as sugars, as well as nucleotides, as well as everything else that is capable of ionization, has a degree to which they will tend to ionize or deionize. In this case, we're talking about proton exchanges. So the ionization is a deprotonation, and the deionization is a protonation, right? So something is, in this example, we have phosphoric acid has a neutral charge. It becomes charged or ionized with a -1 charge if it loses one proton. That is the deprotonated state. Phosphoric acid is the protonated state because that proton is added. The relative degree to which they will tend to lose or gain a proton is a matter of probability, and the probability depends on the concentrations of substrates, of reactants and products. Because H plus is a reactant or product, depending in which way you're going, then the pH, the concentration of hydrogen ions, becomes a critical determinant of the degree to which you would protonate or deprotonate. In general, if it's a very acidic environment, the concentration of hydrogens goes up, and that favors protonation. If the concentration of hydrogen ions goes down, it's more alkaline or more basic, it's less acidic, facilitates deprotonation. We can express the probability that they'll ionize as the pKa. Technically, the K is the equilibrium constant of a reaction. It's saying if you have these two things that can interconvert at chemical equilibrium, when they're both going forward and backward in the same at a constant rate, what would you expect to have more of? The thing on the right or the thing on the left? But if it's a K of an acid, we can call it the K sub A, the K of the acid. And we can then convert that to a pKa, the negative logarithm of the Ka. And the pKa happens to be the pH at which we have 50% the thing on the right and 50% the thing on the left. That's how you think of it technically. But you can more intuitively think about it as if you have two convertible chemical species in a protonated and deprotonated state, they can act as a buffer when the pH of the solution they're buffering is close to their pKa. And the pKa is the pH that they're going to defend. So in this particular case, I took the conversion of phosphoric acid to dihydrogen phosphate, hydrogen phosphate, and phosphate from the Berg biochemistry book. And you can see that their pKa's are 2.12, 7.21, and 12.67. What that means is that you're not really going to see much action between phosphoric acid and dihydrogen phosphate unless you get way more acidic than most of our body tissues would tolerate. And you're not going to see much action between hydrogen phosphate and phosphate until you get way more alkaline than most of our tissues would tolerate. And so what you're primarily going to see as free phosphate in solution is dihydrogen phosphate and hydrogen phosphate. And when they're in solution, they're going to buffer the solution, and they're going to defend the pH of 7.21. If you get less than that, then you're going to favor the protonated form, so it'll absorb hydrogen ions. And if you get higher than that, you're going to favor the deprotonated form, so the hydrogen ions get released. Now, it's also the case that the pKa's are not intrinsic properties of the chemicals, but they depend on the experimental conditions in which they were measured. And that's why if you look at different sources, you're going to see different values for the pKa's. For example, I took the Lehninger biochemistry diagram for the same thing, and they show the pKa as 2.14 instead of 2.12, 6.86 instead of 7.21, and 12.4 instead of 12.67. The pKa's that I'm going to follow are taken from a paper. They're not the ones on the screen. The ones that I'm primarily going to use in the rest of this presentation are taken from a paper by Robergs that I'll show you later, where the most recent pKa's under the conditions that are most relevant to those that prevail in skeletal muscle were compiled from the existing literature. These pKa's that are on the screen are from that paper. What I want to show you now is that the pKa of the phosphate will be radically different depending on what it's bound to. So as free phosphate, the ability of phosphate to bind to one proton is rated at a pKa of 11.59. But when you have glucose 6-phosphate, the pKa of the phosphate group to bind one proton drops down to 6.1. That means the glucose has made that phosphate tremendously more acidic than it was on its own. Why is that? Because the carbohydrate molecule is rich in oxygens, and it has an acidifying effect because of the electronegativity of the oxygens. If you look at ATP, you're going to see considerably higher pKa's, but they're still way lower than the phosphate, but they're higher than glucose 6-phosphate. So ATP is 6.48, ADP is 6.38, and AMP is 6.2. And what you're seeing here is the primary influence is the acidifying effect of the ribose, but it's slightly mitigated by the basic effect of the nitrogen-rich adenine that's behind it. But then the effect of the ribose is mitigated by the distance of the terminal phosphate from the ribose molecule, because it's only the terminal phosphate that's going to tend to be able to bind to even one proton. All these other ionization sites are fully ionized in the phosphate moieties that are bound to ATP. So in ATP, the terminal phosphate is fairly far away from the ribose, but it's closer in ADP because you've chopped off one phosphate, and it's a lot closer in AMP because you've chopped off both phosphates. And so what you see is the acidifying effect of the ribose becomes stronger and stronger as the terminal phosphate gets closer to it. The reason this is so important is that we're going to see that it's not just about the stoichiometric balance of the covalent changes to the molecules in the pathways. It is also about the relative pKa's of the ionizable groups of all the different compounds involved. So Robert Robergs is, if George Brooks has for decades been the lone voice crying in the wilderness that lactate is not a useless and harmful waste product of metabolism, Robert Robergs from Queensland University in Queensland, Australia, has been the lone voice crying in the wilderness for the last decades, saying that lactate is alkalinizing, your acidosis is coming from somewhere else. His papers and what I'm telling you now, in fact, indicate that there's essentially no such thing as lactic acidosis. You can have hyperlactatemia alongside and in parallel with metabolic acidosis, but the lactic acid's not getting made in the body and it can't cause lactic acidosis. All right. So Robert Robergs has done the best work to date in compiling the relevant factors. And one of the things that he's pointed out, he's not the first to point this out, but standing on the shoulders of giants before him, outlined the effects of competition between positively charged minerals and hydrogen ions. He's the first to integrate these competitive ion effects with how they would influence all of the different contributions to acidity in glycolytic energy metabolism during high intensity exercise. And so what you see in this table is he's showing you the different ATP complexes that can form. It can bind to one hydrogen ion. It can bind to 2, 3, or 4. It can bind to one magnesium. It can bind to 2 magnesium, and it can get one proton added with one magnesium, or it can bind to potassium, or it can bind to sodium. And most of these are very small. If you look over here where you see exponential notation, you're talking about super low quantities. Although if you're talking about exponential notation to the -2, you're talking about, it's not that small. You're moving a decimal point over 2 places. But in general, the predominant effects are where you see these single-digit decimals. In the non-competitive column, this is what happens if you just look at the pKa. So the pKa of ATP implies that it should be 23% protonated at pH 7.0. But if you take into account the effects of magnesium, potassium, and sodium, then you come down here and you see that it is 3% protonated at 7.0. Then, of course, this changes as you go down in pH. So a resting muscle cell is going to be pH 7 to 7.2, and in exercise, you could easily get down to 6.7, and you can get down to 6.2. So as you go down to 6, this is kind of the limit of where you could see high-intensity exercising skeletal muscle. And you see that 75% protonation of ATP is only 24% when you take into account the other cations. But 24% is a lot higher than 3%. So there's still a huge difference with pH. It's just that it's nowhere near as protonated as you would expect from looking at the pKa. All right, now here we have all the different species that free phosphate can form. And we can see it can bind 1, 2, or 3 hydrogens. It can bind magnesium with 1 or 2 hydrogens, potassium with 1 or 2 hydrogens, 2 potassium and 1 hydrogen, 2 potassium, sodium and hydrogen, sodium and 2 hydrogen, 2 sodium and 1 hydrogen. So when you take into account all these different possibilities, it looks at first like phosphate would be 36% protonated at 7.0. But when you look at the net effects, the fact that so many mineral bindings can actually exist in protonated form winds up allowing this to bind an average of 1.3 protons. Of course, this goes up quite a bit to 1.8 protons when you go down in acidity. Now, the reason this is important is because if you're removing phosphate from a solution, then depending on the balance of cations and the pH and so on, you could be removing 2.6 protons. You could be removing the phosphate that is binding 2.6 protons if you remove 2 phosphates from solution in the GAP-DH reaction of glycolysis, where 2 molecules of glyceraldehyde 3-phosphate and 2 free phosphate and 2 NAD wind up producing 2 NADH and 2 molecules of 1,3-bisphosphoglycerate. Now, the fact that it can go up to 1.8 at pH 6 seems to imply considerable buffering capacity, but the demands of covalent reactions in glycolysis can override that. For example, in the GAP-DH reaction, if you need PO4, you're not taking 2 extra hydrogen ions along for the ride. So it doesn't matter if the phosphate has buffered additional protons. If you're going to take that phosphate away, use it in the reaction, and throw away all the protons that had bound to it. So what you see is that on the one hand, the free phosphate has buffering capacity where it gets more protonated in the more acidic state. But on the other hand, certain reactions in glycolysis are going to become more acidifying when they use that phosphate, because all the work it did to bind up extra protons gets thrown in the trash when that phosphate gets entered into a chemical reaction. All right, so now let's go in order. Let's look at the reactions of glycolysis and just put them in order to see how things are stringing together. The two phosphorylations at the beginning are going to each release a proton. That's 2 protons. This is the Berg biochemistry textbook showing the glyceraldehyde 3-phosphate dehydrogenase reaction. It shows you that NAD plus becomes NADH and H plus. This happens twice. So it shows you the addition of 2 more protons. We're at 4 total now. What it doesn't show you, though, is the organic chemistry of the phosphate reaction. It totally leaves this unexplained. But that phosphate was coming from what was primarily hydrogen phosphate, a little bit of dihydrogen phosphate, right? An average of 1.3 protons carried by that phosphate. Those protons are not present on the 1,3-bisphosphoglycerate molecule. That means they too are released into solution, right? So this is the NAD is stoichiometric. It for each NAD plus, you're going to get NADH plus H plus. It's going to 1-to-1 even out like that. But there's a very large non-stoichiometric fractional probability-based release of an average of up to 2.6 protons by removing the phosphate from solution. So technically we're not up to 4, we're up to 6.6. Now for the phosphoglycerate kinase reaction, the Berg biochemistry textbook does show it consuming a proton, but offers no explanation for where that proton gets used up. You can see that there's a hydrogen here, there's 2 hydrogen here, there's 1 hydrogen here, and they're all exactly the same. So this H plus disappears without ever showing where it went, and you're left to kind of assume that it got soaked up into the ATP molecule, only it didn't, because as I showed you before, mechanistically, the substrate level phosphorylations transfer PO3. There's no additional oxygens involved. There's no hydrolysis, dehydration, synthesis. There's no reason this H plus should get sucked up into the ATP molecule. And in fact, the Lehninger biochemistry textbook, consistent with the other reaction I showed you, is more clearly demonstrating the organic chemistry of the phosphate transfer, and it shows you that it's PO3 for PO3. There's no hydrogen involved. So the Berg biochemistry textbook is actually giving you an extra hydrogen ion, not explaining it at all, but by the organic chemistry of the phosphate transfer, it doesn't exist. Now, this is what actually happens for why a proton does get consumed in pyruvate kinase. As I indicated before, that hydrogen is becoming the 3rd hydrogen of the last carbon. You're taking that phosphate away and you're leaving behind the oxygen, and that oxygen is going to want to pull in a hydrogen if it can get one, because a COH bond is not usually going to ionize. But as it so happens, because of the enol ketone tautomerization that I showed you before, you wind up making that double bond transfer up here. You form a ketone carbonyl, and you bring the hydrogen down here to make the terminal methyl group. You're almost never going to have, a carbon-carbon bond having a hydrogen ionize off of it. So that is why you're absorbing the proton, the pyruvate kinase reaction. When Lehninger shows the pyruvate kinase reaction, unlike the Berg biochemistry textbook figure, which states out, I showed you the Berg textbook figure earlier. So the Berg pyruvate kinase reaction is shown up here, and it shows you it doesn't really clearly explain where it goes, but it shows you the proton coming in that winds up making up that methyl group at the end. The Lehninger textbook does not show you this at all. So it shows the PO3 transferring, and you have a CH2 here, you have a CH3 here, and nothing in the text and nothing in the figure explains where that extra hydrogen came from. But It came from drawing a proton out of solution, which had an alkalinizing effect. Any textbook will acknowledge that the pyruvate to lactate conversion converts NADH plus H plus to NAD plus for all the reasons we've already described. The lactate dehydrogenase reaction is intrinsically alkalinizing. For the purposes of our lesson here, we're going to consider lactate always the end product of glycolysis. These two papers will tell you much more about that. One is called Lactate is Always the End Product of Glycolysis from 2015 from Rogatsky et al. And one from George Brooks et al. Tracing the lactate shuttle to the mitochondrial reticulum in 2022 compile abundant evidence that lactate is the default end product of glycolysis. So if we look at these textbook descriptions, we see that the Berg biochemistry textbook does show us 2 protons released in the initial phosphorylations, 2 released in the reduction of NAD+, 2 consumed in the pyruvate kinase reaction, and it wrongly shows us that 2 are consumed in the phosphoglycerate kinase reaction. So according to the graphics in the Berg biochemistry textbook, there are net 0 protons going from glucose to pyruvate. Yet in the stoichiometry, it ignores all of those and only shows a yield of 2 protons that came from the conversion of 2 NAD plus to 2 NADH. It completely ignores all of the other proton transactions that it showed in the graphics of glycolysis. Now, Lehninger shows the exact same stoichiometry, and it's somewhat more self-consistent because it never shows you the proton exchanges in the reactions except for the conversion of NAD plus to NADH. But Lehninger does show you in the graphics that there are phantom magical appearances and disappearances of hydrogens in the molecule and never accounts for how they got there or where they went. And so none of this is to say that the biochemistry textbooks, that the authors aren't good enough to do this work. It seems to me, I do think there's a mistake in the Berg phosphoglycerate kinase reaction, but it just seems like they don't consider the acid-base balance of the reactions to be important enough to fully account for the protons, and so they just often leave it out. But that leaves the student in a pretty bad position because you think that's your go-to source when you want to, a light bulb goes off in your head and you say, oh, I wonder where the acidity is coming from. And you go to the textbook and they show you some of the protons and they don't show you other protons and they don't tell you what happened to them and they don't put it in the stoichiometry. And so once you want to start understanding a question like, where does acidity come from in skeletal muscle glycolysis, you're left needing to go way beyond the textbook to get anywhere when you want to answer a question like that. And I think that's unfortunate because it's not a whole lot of extra effort to show the proton transactions. All right. So that's where we get the net acid-base balance in glycolysis from. And that's why the production of lactate is intrinsically alkalinizing. But this is why we know that we do not ever make lactic acid. These figures are from the Berg biochemistry textbook, and they show you on the top the glyceraldehyde 3-phosphate dehydrogenase reaction split into 2 half reactions, and then on the bottom is the phosphoglycerate kinase reaction. So in this first half reaction, they're depicting this hydrogen, shown in blue, that is being added to NADH. You then have water, which has an OH group that gets added in its place, constructing the carboxyl group, and then the proton that is left over from H2O is the proton that's released into solution. Before that carboxyl group could ever deprotonate or do anything of the sort, we are switching it out for a phosphate group, and that's going to happen through dehydration synthesis. But remember, phosphate is mostly complexed as hydrogen phosphate in solution. And so this dehydration synthesis is taking a hydrogen from the phosphate and it's taking OH from that molecule, and that's coming out as water, and the phosphate's getting added there. No net transactions of protons, no carboxyl group ionizing. And then in the phosphoglycerate kinase reaction, we have Berg wrongly showing us the proton here. But what's most important is the PO3 is coming off, being added to ADP to make ATP. And what did you do? You took it off, leaving behind the negative charge. So this is the carboxyl group formed in the ionized state, never protonated, never deprotonates to form an acid. You did not form 3-phosphoglyceric acid. You formed 3-phosphoglycerate. That 3-phosphoglycerate became 2-phosphoglycerate, phosphoenolpyruvate, and pyruvate, and then lactate never became 3-phosphoglyceric acid or phosphoenolpyruvic acid or pyruvic acid. We're not saying lactate, pyruvate, glycerate to save syllables. We're saying lactate, pyruvate, glycerate because these are formed and stay in their anionic, negatively charged, deprotonated form and never contributed a proton to solution at any point. And you can look at the carboxyl group from the point where it is first formed in the phosphoglycerate kinase reaction, and you can carry it through all the other reactions, and you can watch that carboxyl group stay ionized right up until you get to lactate. So the human body does not make lactic acid. It never makes lactic acid. Now, what is the fate of that lactate? So as extensively documented by George Brooks, especially in that article tracing the lactate shuttle to the mitochondrial reticulum, we have a cytosolic lactate dehydrogenase that is converting 2-pyruvate to 2-lactate and oxidizing 2-NADH to 2-NAD+. It's going to travel through the voltage-dependent anion channel, or VDAC, in the outer mitochondrial membrane into the intermembrane space of the mitochondrion. We're kind of in the intermembrane space. We're kind of in the cytosol, and that's because the VDAC is very promiscuous about what it transports. Lactate, pyruvate, NADH, NAD+, hydrogen ions. Any of that stuff gets back and forth through the VDAC, whereas the inner mitochondrial membrane, which guards the entry to the matrix, which is the very inside of the mitochondrion, has very specific channels in its membrane that are very discerning in what they let in and out. And that makes the mitochondrial matrix very different in composition from the intramembrane space or from the cytosol, whereas because of the promiscuous nature of the VDAC, the cytosol and the intramembrane space are going to be fairly consistent in their composition with one another. When the lactate comes in here, it then can be converted back to pyruvate by mitochondrial lactate dehydrogenase. Now, lactate dehydrogenase converting pyruvate to lactate releases energy. Lactate dehydrogenase converting lactate back to pyruvate incorporates energy. It's hard to do that without an energy source. So what you find is that mitochondrial lactate dehydrogenase is physically connected to complex 4 of the respiratory chain. And the heat that is released when oxygen is converted to water in complex 4 is funneled into the mitochondrial lactate dehydrogenase reaction to convert the lactate back to pyruvate. And then the 2-pyruvate enter the mitochondrial matrix through the mitochondrial pyruvate carrier, where they can be further oxidized in further reactions. Now, when you do that, you convert the NAD back to NADH plus H+. So you've reversed the alkalinizing effect of the cytosolic LDH reaction. However, you've done two things. One is that NAD plus is needed to keep glycolysis going. You have moved the NADH over here. Even though these can go back and forth in the VDAC, now you're way over here, and you've got to take some time to get back over here, right? In the proximate vicinity of glycolysis, you've gotten rid of NADH, you've kept NAD plus there, and you've gotten rid of the acidity that's going to inhibit glycolysis. You've moved it over here. More to the point, if the respiratory chain is actively running, that NADH is going to be quickly converted to NAD plus, and then you've preserved the alkalinization. You've preserved the provision of NAD plus, and it doesn't matter that these can get back here because they're going to be very quickly oxidized, retaining the alkalinity and retaining the redox reactivity because of the respiratory chain's activity. If lactate accumulates in muscle, it's going to be because the oxidation of the NADH by the mitochondrial respiratory chain is not keeping up with the degree of glycolysis. But because the mitochondrial respiratory chain is one of your chief alkalinization strategies, then that also means that accumulating lactate is going to be a defense against the rising acidity that would occur under those conditions. An alternative fate for lactate would be to leave the cell, and it can leave the cell to a nearby cell, or it can leave the cell into the blood. If it leaves the cell from a fast twitch muscle fiber to a slow twitch muscle fiber nearby, you're never going to see it rise in the blood, and it's going to get oxidized in the slow twitch muscle fiber. If you exceed the capacity of the slow twitch muscle fibers to do that, you're going to spill it into the blood. It's going to get taken up by the liver. That's called the Cori cycle, and it's going to be converted into glucose. Either way, you have removed 1 proton per lactate molecule from the muscle cell, because lactate transport goes through a series of transporters in the monocarboxylate transporter family, or MCT family, which transport things that have 1 carboxyl group, such as lactate or pyruvate, etc. And they are H+ symporters. Symport means transport in the same direction. Antiport means transport in opposite directions. These are proton-coupled symporters, which means 1 for 1. Lactate leaves, H+ leaves. So if 2 lactate accumulate at the end of glycolysis and then leave the cell, they carry 2 protons with them. So not only did the lactate dehydrogenase reaction remove 2 protons into the NAD+ formation, the lactate molecule transported 2 protons, or 2 lactate molecules transported 2 protons out of the muscle cell. So you've now doubled the alkalinizing effect of the lactate dehydrogenase reaction, and you've tripled your net alkalinization of the alkalinizing phase of glycolysis. This is from a graduate student in Robert Robergs' lab. So Robergs was at this 23 years ago. They say the results of our study showed that hexokinase drops pH by 1.52. The glyceraldehyde 3-phosphate dehydrogenase reaction coupled to the phosphoglycerate kinase reaction drops at 0.43. And the ATP hydrolysis reaction drops at 0.93. So these are all acidifying reactions. Whereas the pyruvate kinase reaction raises the pH 1.9, and the LDH reaction raises the pH 3.1. Raising the pH 3.1 units is 1,000-fold alkalinization. So they conclude down here, these results confirm that lactate production does not cause metabolic acidosis, and that biochemical contributors to the development of acidosis include glycolytic flux, NADH plus H plus accumulation, and ATP hydrolysis. I mean, what an understatement. Lactate production does not cause metabolic acidosis? These results indicate that lactate production makes you 1,000-fold more alkaline. Now, granted, these are in vitro conditions, and Robergs has since done a lot of modeling that includes lots of empirical data in humans with metabolites and in vitro pKa's and competitive cation binding, where I think the modeling is very superior to the in vitro what happens when I just take this isolated enzyme in solution that was done here. But this provides proof of principle that the early glycolysis reactions are acidifying and pyruvate kinase and LDH are alkalinizing. In 2017, Robergs published a paper that in PLOS1 that compiled all the different reactions of non-mitochondrial energy metabolism, focusing on glucose metabolism and the phosphagen system. And what you can see here is that the delta H shown on the right is the stoichiometric gain or loss of an electron as a result of the covalent chemical reactions taking place at those steps. So a couple things to note is that creatine kinase helps mitigate the loss of ATP, but that is the same as if you synthesized ATP in the mitochondrial respiratory chain. So the creatine kinase reaction is removing 1 proton from solution. The AMP deaminase reaction, which is what happens when you start to get down to AMP and then you start breaking it down further than that, ultimately that could go down to uric acid. That also is removing one proton from solution. You're going to be doing a lot more of creatine kinase than AMP deaminase. ATP hydrolysis releases one proton. The phosphorylation of glucose and fructose 6-phosphate each release one proton. The glyceraldehyde 3-phosphate dehydrogenase reaction, GAPDH, it releases one proton. But remember, not shown here is the non-stoichiometric net effect of removing phosphate from solution. You're removing buffering capacity, and you're releasing double the number of protons as you are when you just do the stoichiometry. And then the pyruvate kinase reaction is going to remove one proton, and it looks like this got cut off on the screen, but lactate production is also going to remove one proton. This has it all incorporating the pKa's and the competitive cation effects of the relevant reactions, and that then adjusts what you would expect based on the stoichiometry alone. And that means that the AMP deaminase reaction is a little bit more alkaline than you would have predicted based on the stoichiometry. ATP hydrolysis is a little bit less acidifying than you would have predicted. Creatine kinase is basically right on the money at pH 7, but it starts to lose its power slightly at pH 6. And ATP hydrolysis at pH 7 is significantly acidifying, but that effect completely disappears by the time you get to pH 6. And that's a result of the fact that as you start hydrolyzing a lot of phosphate, the buffering capacity of the phosphate starts picking up. Now, on the other hand, the reaction that uses the free phosphate and removes it from solution, because it's removing a buffer from solution, it's becoming a lot more acidifying at pH 6 than it is pH 7. So pH 6 is the light bars, pH 7 is the dark bars. So you can see the GAPDH reaction is in net, almost one proton releasing at pH 7, but it's more than 1.5 proton releasing when you get to pH 6. Then you can identify a number of other reactions. So one that we haven't really talked about is that there's a slight acidifying effect of the phosphorylase reaction that breaks apart glycogen. But when you use the phosphorylase reaction, you get to glucose 1-phosphate, and you don't have to use the hexokinase reaction. The hexokinase reaction is much more acidifying than the phosphorylase reaction, and that's why going from free glucose through glycolysis instead of coming from glycogen leads to more acidity. But that's very mitigated at pH 6. The phosphofructokinase reaction is considerably acidic at pH 7. It kind of disappears at pH 6. And then when you get to phosphoglycerate kinase, you have an interesting effect here where 1,3-bisphosphoglycerate, which came out of the glyceraldehyde 3-phosphate dehydrogenase reaction, has a very high ability to bind protons. But 3-phosphoglycerate, which comes out of the 3-phosphoglycerate kinase reaction, does not. And so even though you're not doing a proton transfer in this reaction, you are in net changing a substrate from one that binds protons pretty tightly to one that does not bind them tightly. And so you do have net acidification here. And then you can see pyruvate kinase is substantially alkalinizing at pH 7, and it starts to lose that effect at pH 6. And lactate dehydrogenase is pretty much always 1-to-1 alkalinizing. So lactate dehydrogenase is always your most alkalinizing reaction, and it never really changes, even as the pH drops to 6, which is beyond the maximum you'd see in exercising skeletal muscle. The lactate dehydrogenase stays the most alkalinizing reaction. When he modeled the effect of glucose to pyruvate versus glucose to lactate along pH, one thing that you can see is glycogen to lactate is always slightly more alkaline than glucose to lactate. Glycogen to pyruvate slightly more alkaline than glucose to pyruvate. As you go down in pH, you unfortunately wind up with glycolysis becoming much more acidifying. And so that kind of explains why as you approach muscular failure, you get closer to it instead of further away, right? You don't have intrinsic buffering capacity. You're burning through more glucose to try to get that last stretch of ATP. And as it becomes more acidifying, as it becomes more acidic in the muscle, that doing more glycolysis gets more acidic, which it's like the closer you get to failure, the closer you get, and then you fail. And then the other thing to notice is that, when you go to lactate, you're way more alkaline than when you go to pyruvate. And this reflects the fact that making lactate is highly alkalinizing. In this next paper, Robergs took data from 25 human exercise trials that provided the time course of metabolite accumulation during exercise at relevant intensities, and then the data from biochemical reactions, pKa's, competitive ion effects, etc., from the 2017 paper, slightly updated, and then put these all into a model that said, okay, if you have a hypothetical exercise session that is totally anoxic and hits failure at the 3-minute mark, what would be the time course effect of the production of acidity from different sources? And what you can see here is that at the very beginning, the creatine kinase reaction slightly outperforms ATP hydrolysis. Creatine kinase is alkalinizing, ATP hydrolysis is acidifying. You'll see that like in the first seconds of muscle contraction, the pH goes up a tiny, tiny bit, and then it starts falling. And at first, ATP hydrolysis is the powerful acidifier, but this gets replaced by glycolysis as soon as glycolysis kicks in. And the effect of ATP hydrolysis really starts to disappear as you get to the 1 to 1.5 minute mark, because ATP hydrolysis becomes net pH neutral at lower pHs due to the proton binding capacity of free phosphate. And so what takes over as a source of acidity is early glycolysis, but especially the GAP-DH reaction, which is always your most acidic reaction. And as you get to failure, you're not changing the pH anymore once you stop contracting. On the top, you see the alkalinizing factors, and as creatine kinase starts to fade away, the later glycolysis, pyruvate kinase and LDH reactions take over, and LDH is always your most alkalinizing reaction, and it starts to really exceed the pyruvate kinase reaction by a margin as you get deeper into exercise towards the failure point. They did not include hexokinase in this model because they were modeling it from glycogen. To me, when I look at these papers, it looks to me like, yes, glycogen is dominant, but you still have a lot of hexokinase going on. The limitations that they acknowledged was that this is a specific extreme model of anoxic environment, 3 minutes to failure, that's not reflective of a lot of different types of exercise. It's one thing to model that this is where H plus is being generated. It's another thing to say that this is the cause of muscle pH. In order to prove cause and effect, you'd have to intervene to stop one of those reactions and show that that alkalinizes the muscle, or to stop LDH reaction, show that that acidifies the muscle. So they're not showing the cause and effect behind the pH. It's still up for debate. They're modeling the different sources of an isolated set of things that can generate protons or remove them. They did not model lactate transport, which would provide very powerful additional alkalinizing effects, because they said there's not enough information on the causes of the magnitude, the quantitative magnitude of the transport and what causes it to vary under different conditions. And the alkalinizing effects of the respiratory chain were totally ignored, which are the dominant alkalinizing effects until your glycolysis starts exceeding its capacity. I would add additional limitations that they didn't look at glycerol 3-phosphate. I'm going to show you that. And I also think, I extracted all 25 of these studies to look for evidence of where could you see where glycolysis was getting backed up? Was all of glycolysis running at the same time, or were you stopping at a certain point? And if you were stopping at a certain point, was it at GAP-DH because you ran out of NAD+, or was it at PFK because you got too acidic? What I found was very few of those studies showed you the difference between those different stopping points in glycolysis. There's tremendous amount of studies that measure lactate or ATP, ADP, creatine phosphate. There are not many studies that look at fructose 1,6-bisphosphate. There are, even I found one study, and it wasn't included in the 25, that looked at any representative of lower glycolysis after the GAP-DH reaction before pyruvate. I found one study that was not included in this model. So I do think that this model is limited by the very limited number of studies that are actually looking granularly at the different points of glycolysis. And so for that reason, I did my own analysis just looking at the individual studies to see what additional points they tell us. And we'll get to that in a moment. But for now, I want to take a brief break from the biochemistry of lactate and just say, okay, what do we know about fatigue? And this is a great diagram that was tweeted by Chris Beardsley of Strength and Conditioning Research, where he listed the mechanisms of fatigue. Like if lactic acid doesn't cause fatigue, what does, right? And so he he reasoned the effect on ATP in the second column, because the point that he wants to make here is that you don't run out of ATP, but you have a lot of fatigue mechanisms that prevent you from running out of ATP. And so you could regard a lot of these as what is going to happen when the body is in danger of running out of ATP, but it needs to make sure it doesn't. But as we go through them, you'll see that some of them are actually, damage is happening. It's kind of an accident that it's conserving ATP, and some of them might be purposeful regulation. So coordination disruption is when the ability of the muscle to keep up starts dropping, and the brain does not step up its game to compensate and make the muscle keep going. Supraspinal CNS fatigue is when discomforting sensations increase the perception of effort and make you want to stop. It's eroding your willpower. Spinal CNS fatigue is when motor neurons of the spinal cord desensitize to the amount of the brain telling them what to do, and they start to get less sensitive to that, so they transmit less signal in muscle. Loss of cell membrane excitability is really about like your energy capacity is going down and it's not properly pumping out the ions. So your resting potential in a neuron is built on the ion gradients. And if they all get let in through an ion channel to activate the neuron, they got to get pumped back out. So if you're running into a problem where you're going to run out of ATP if you use enough to keep the neuron excitable by pumping the ions back out, then you're between a rock and a hard place in that if you conserve that ATP, you are going to be less effective at pump. I guess it's not a rock and a hard place. It's just you're at a point where you have to lessen the muscular contraction by one way or another. One is you run out of ATP, and the other is that you conserve it by not pumping the ions out of the neuronal membrane as well. And if you don't pump the ions out to conserve your ATP, you're going to have the neuron less excitable, and it's going to be less stimulatory to the muscular contraction. You can have damage from proteases that get activated by calcium ions. Calcium ions are released as a signal to contract muscle. They're released as a signal to release neurotransmitters, and they have to be energetically pumped back into their place. And so if you are losing energy to control the calcium ion distribution, they can activate proteases that start causing damage to the membranes that are controlling the coupling of neural activity with muscular activity. You can lose sensitivity to the calcium ions because you are just, too much calcium ion signaling leads to becoming tolerant of it and desensitized to it. That makes you contract muscle less. And then finally, you have accumulation of phosphate and hydrogen ions. Here's our acidity. So it's a major mechanism, but it's one of 7 major mechanisms that he's highlighting. And the hydrogen ions are not caused by lactic acid, but nevertheless, it is a mechanism of fatigue. So we do have to understand where it's coming from. one of the hypotheses that Robergs was not covering that I've seen in the literature, and I took this from a paper called An Obsession with CO2. And this is the CO2 hypothesis of acidity. And so I looked at some of the papers that were cited in favor of it. And this is a paper where they did 30 seconds of maximal exercise on a bike. And 6 people, they took muscle biopsies. 3 people, they took blood samples. And they did the exact same protocol. And they put them all in one table to compare them. So one thing that you can see is that if you look at venous plasma, the lactate, and milliequivalents per liter is the same as millimoles per liter. The lactate goes from 1 to 13, so it increases 13-fold. The muscle lactate goes from 6 to 47. Now, on the one hand, it's not as great as a fold increase. It's 7.8-fold instead of 13-fold. But on the other hand, the lactate concentration in the muscle gets way higher than the concentration in the blood. And like I said before, I don't think, or according to Robergs, we don't know that much about what dictates the quantitative transport. We do know that it goes through the MCT, which is a proton symporter, but we don't know, what dictates why the muscle would transfer it at X rate. You can imagine easy solutions to this, right? If lactate transport removes protons from the muscle at the expense of acidifying the blood, then presumably maintaining blood pH within narrow boundaries that are consistent with life is more important than getting the protons out of the muscle, especially when the muscle can tolerate more acidity than the organism can in the blood. You can just accumulate high concentrations of lactate and absorb protons that way, where the protons are actually they're actually being removed. The total number of protons is decreasing when you accumulate lactate. It's just moving from one compartment to another when you transport lactate into the blood. So, presumably I would think that this is going to be limited by the fact that you can only transfer protons into the blood at the rate that the blood can tolerate, but you can accumulate lactate, which has less of an alkalinizing effect on the muscle, but costs less to the rest of the body. And so I think the muscle is going to seek a balance between those two priorities. And that's why you have, considerable increase in the blood and yet really massive accumulation of high levels in the muscle cell to reach 47 millimoles per liter lactate. You can also see that muscle CO2 goes from 46 to 106 torr, and I tried to calculate that out to millimoles per liter, and I get that that's 1.41 to 3.25 millimoles per liter, and that's increasing 2.3-fold. And the hydrogen ions increase 2.5-fold, which is pretty close to what the CO2 is doing. So I can see someone looking at this and I can see them saying, well, the acidity is really following the CO2. CO2 goes up 2.3-fold. Hydrogen ions go up 2.5-fold. CO2 is mostly the acidity because CO2 is acidic. Why? Because CO2 dissolves in solution. It becomes hydrated. It becomes carbonic acid that dissociates a proton to become bicarbonate. Well, here's the problem I have with that. That is that the bicarbonate goes nowhere. So the bicarbonate is going from 9 to 7 in the muscle, and it's going from 27 to 28 in the blood, right? So the CO2 is what's acidic. How is it becoming acidic if it's not ionizing to bicarbonate, right? And I think, the explanation is pretty simple. You absolutely need to go back and forth between CO2 and bicarbonate to get CO2 from the muscle cell to the lungs. But that happens extremely rapidly with the carbonic anhydrase reaction, 13,000 times higher than spontaneous. And CO2 rapidly crosses cell membranes, and it only takes 5 seconds to get from your muscle cell to your breath. So yeah, the CO2 partial pressure is increasing, but that's just representing a 5-second lag to get out the breath. Okay, on the other hand, they published a study, same protocol. It's probably the same study as common in when you do small studies, you want to get more papers published. So you take one study and you make 3 papers out of it. So I think that's what they did. But they showed that in another paper that CO2 lagged by minutes, right? So it's immediately it's coming in the breath, but it stays high for a few minutes. You're not exercising anymore. Why is that? So they reasoned that the CO2 is being stored in the muscle, but it wasn't being stored in the muscle as bicarbonate. So it must have been stored some other way. And they suggested it was forming carbamate ions on the amino groups of proteins and amino acids. And that is a 1-to-1 release of a proton per CO2 molecule. So if carbamate is forming and is accounting for some of the acidity, you got to compare it to the other possible sources. So how do you get CO2? It's after you get to lactate, you convert it back to pyruvate in the intramembrane space. You oxidize the pyruvate with pyruvate dehydrogenase, and you enter the citric acid cycle, and you run the citric acid cycle, and all of that is facilitated by running the respiratory chain. By the time lactate's accumulating, by definition, you are not making CO2 out of the lactate. And that's not to say that you're not making CO2 anymore. It's just to say that the marginal increase in lactate is reflecting the marginal increase of glycolysis that exceeds the capacity for the respiratory chain to meet that marginal increase. And the acidity rises when the respiratory chain can't keep up. So if the acidity is going up in parallel to the lactate, that is reflecting the margin that does not generate CO2, that is acidifying because the respiratory chain could not step up any higher to get more CO2 out of that lactate. So you have to realize that by the point that the acidity is happening, it's happening in parallel to you not making CO2 to meet the level of glycolysis. So it's pretty unlikely that CO2 is accounting for the increase in acidity that happens when the respiratory chain can't keep up. But if we just logic our way through it, if you fully oxidize the glucose in the respiratory chain, you'd get 6 CO2. If 6 CO2 are 10% forming carbamate, that's 0.6 CO2 per glucose molecule. By contrast, in the first half of glycolysis, you can generate 6 protons. So, that's 10 times the theoretical yield from the acidifying phase of glycolysis as you could get from carbamate, even if you were fully oxidizing it. But again, you acidify when you are not fully oxidizing to CO2. So let's look at 3 individual studies, and we're going to focus on this first one. 8 healthy men performed an isometric contraction on a knee extensor machine at 2/3 maximal force. They took biopsies at rest, 20 seconds into the contraction, and at the point of fatigue. All right, so I've put the metabolites that they've measured along with the concentrations at rest at 20 seconds and at fatigue, and then I calculated the fold increase at 20 seconds and the fold increase at fatigue. And I drew out the pathway here where some of these grayed out steps indicate that they didn't measure that, but it is how you get from one to the other that is important to draw attention to. The first thing to note is that the lactate is the thing that's most increased. The second most increased point is glycerol 3-phosphate. We have not talked about that yet, but glycerol 3-phosphate accumulates as a side reaction when DHAP does not get metabolized through GAPDH. So if NAD is not abundant and the GAPDH reaction doesn't go forward, then DHAP accumulates. But if NADH is abundant and DHAP is abundant, then that will drive forward the reaction to form glycerol 3-phosphate. I believe the primary drivers of this reaction going forward are going to be the concentration of DHAP and the high NADH to NAD plus ratio. I do think it's possible that there is some influence of pH, because based on the little research that I did, it looks like the pH optimums of these enzymes are a little bit different, such that acidity is going to favor the production of glycerol 3-phosphate. But I think it's going to be mostly driven by the NADH to NAD plus ratio. And to some extent also the degree to which the influx is inhibited or not, right? So the influx into DHAP is high and the NAD plus is low and the NADH is high, you're going to get a lot of DHAP and a high NADH to NAD plus ratio, and that's going to drive DHAP into glycerol 3-phosphate instead of down through glycolysis. It's also the case that glycerol 3-phosphate is able to deliver electrons to the respiratory chain through what is called the glycerol 3-phosphate shuttle, which is using an enzyme called mitochondrial glycerol 3-phosphate dehydrogenase. And it does make sense that if the flux through glycolysis expands in exercising skeletal muscle, you're going to need to have more glycerol 3-phosphate in order to turn over those electrons, because it's not disappearing when it does that. It's cycling, right? So you will need some more glycerol 3-phosphate, but you only need enough to accommodate the flux through the pathway. And if you look at glycerol 3-phosphate being 28.7-fold increased compared to the metabolites on either side of it being 2 to 3-fold increased, then you're around an order of magnitude higher than you need to be to accommodate the flux going through the pathway. So clearly this is not a reflection of the use of the glycerol 3-phosphate shuttle primarily. It's a tiny bit that, and it's mostly a side reaction to generate NAD plus and to get rid of acidity. It also appears that there is inhibition of the phosphofructokinase reaction, or PFK reaction, which converts fructose 6-phosphate to fructose 1,6-bisphosphate. Why do I say that? Because if you look at glucose 6-phosphate and fructose 6-phosphate, they're both way more accumulated than fructose 1,6-bisphosphate. In fact, I added them up and I took the ratio, and there's 6.7-fold greater accumulation of hexose monophosphates, that's glucose and fructose 6-phosphates, than there is fructose 1,6-bisphosphate. So I would say the first thing that's happening here is lots of lactate production. The second thing that's happening here is lots of glycerol 3-phosphate production. And the third thing that's happening here is a considerable amount of PFK inhibition. Like I said earlier, PFK is strongly inhibited by acidity. And so you're responding to the fact that the GAP-DH reaction is the most acidic reaction in all of glycolysis. So you don't want to press it forward under conditions of acidity. In fact, as Robergs has shown, it gets more acidic the more acidic you are. So running GAP-DH is a vicious spiral towards never-ending acidity. And you don't want to run it if you're not able to complete the rest of this going forward. In particular, when your respiratory chain being limited is the primary driver of acidity. So there are a number of compensations that are happening here, but it's pretty clear that lactate production, while it's the biggest one, is not capable of fully regenerating enough NAD to keep GAP-DH running forward enough that you wouldn't instead run the glycerol 3-phosphate production. It's also clear that that situation would be a lot worse if PFK weren't inhibited by acidity. And so you have basically a trickle through middle glycolysis, because you are driving the middle into a side reaction and you're inhibiting the top, and you are essentially accumulating acidic hexose monophosphates at the expense of risking running the GAP-DH reaction without being able to get it all down to lactate, because that risk would be running the most acidic part of glycolysis. But that's, it's not all driven by acidity inhibition. It's also driven by collateral damage of the acidity is also the low NAD supply. And that's really what's driving this side reaction. So when I look at this and I look at the Robergs modeling papers, which I think are amazing, I do think that missing glycerol 3-phosphate production is a major missing piece of them. And I also think that PFK inhibition is kind of a wild card where the degree of PFK inhibition might vary in different studies, and that's going to be a really big determinant of how much running gap DH is contributing to acidity. And then I also think that we might be underestimating the hexokinase reaction here. That was kind of left out of the Robergs models, to my understanding, because the assumption is you're coming down from glycogen under these conditions. And I think you are, but you have a lot more G6P than G1P, which I think indicates that even though your fold increase in G1P is almost double your fold, even though your fold increase in G1P is considerably higher than your fold increase in free glucose, almost double. The increase in glucose 6-phosphate, it's hard to say where it's coming from, but it just seems like you could have plenty of hexokinase going on in these conditions. And so when I look at, what are the lingering sources of acidity here, hexose monophosphate accumulation is the most acidic thing that's not being considerably inhibited in all of glycolysis. So I think it's possible, and I'd love to talk to him about it, but I do think it's possible that the Robergs model might be underestimating the contribution that you can get from hexokinase under conditions where hexose phosphates are accumulating, PFK is strongly inhibited, and GAPDH is largely driven around. Now, I couldn't find any other studies that looked at 3-phosphoglycerate or any other reflection of the alkalinizing phase of glycolysis prior to pyruvate. This study showed that glycerol 3-phosphate goes up 23.6-fold during maximal exercise, which I think is consistent with the last study showing that glycerol 3-phosphate production is very important. And this study showed glycerol 3-phosphate went up 12.8-fold during electrical stimulation of contraction. So there is a pretty consistent finding that when these studies look for glycerol 3-phosphate, they do find that it is not as significant as lactate, but very considerable. So what can we conclude? First of all, the primary culprit in acidity is that the alkalinizing respiratory chain is lagging behind acidifying glycolysis. That is number one. Number two, within that context, if you look at the molar sum of glycerol 3-phosphate and lactate, when you start to hit failure, it's 72.4. That's outcompeting the increase in inorganic phosphate of 52.8, which is fairly close to the accumulation of hexose monophosphates at 41.9. So that indicates that NADH plus H accumulation, which is reflected in the glycerol 3-phosphate and lactate accumulation, that's the greatest driver of acidity, followed by ATP hydrolysis, followed by hexose monophosphate accumulation. I was saying before that the hexose monophosphates look like they're the greatest accumulation that's contributing to acidity. What I mean by that is lactate and glycerol 3-phosphate are fixing the problem of NADH plus H plus accumulation, right? The fact that they are so high reflects that the NADH plus H plus accumulation is the primary driver of acidity, but it's also the one that's primarily being fixed by reversing that accumulation in the production of lactate and glycerol 3-phosphate. So what I mean when I say the hexose monophosphates are the ones left standing is that they're the ones that aren't being fixed, right? They're not being cleared, and they are acidic. So I think that's kind of the elephant standing in the room, is that quite often they're left as the one thing that's not resolved that's very acidic. ATP hydrolysis is going to be the driver of acidity that's reflected in the inorganic phosphate accumulation, But Robergs has shown that ATP hydrolysis stops being acidic as you go forward. So I think looking at the inorganic phosphate left over at fatigue is not really how you can measure it. And his modeling indicates that about 25% of the acidity that would be generated across 3 minutes of anoxic contraction to failure is generated by ATP hydrolysis. Carbamate formation may make some contribution, but as lactate rises, this is reflecting the margin at which carbohydrate no longer generates additional CO2, and there really is no hard evidence of carbamate accumulation in the muscle. Like I said before, it's been hypothesized that the delay in CO2 release is a result of stored CO2 in the muscle, but it might just be that once you are not exercising anymore, now you're oxidizing that lactate. And you could be replenishing glycogen, but the point is, it's an open question about the degree to which carbamate formation might be a contributor to acidity in exercising skeletal muscle. The primary defenses against acidity are lactate production, glycerol 3-phosphate formation, and PFK inhibition. PFK inhibition is slowing the input into the highly acidic GAP-DH reaction. So what does this mean for lactate supplements? Andy Galpin tweeted out this 2024 August paper on a randomized controlled trial crossover design looking at a lactate supplement. And, the TLDR of it is they increased the amount of work done by 4% with the oral lactate supplement, and it didn't impact anything else that they measured. If you look at what they did, they gave people a calcium, magnesium, vitamin D3 lactate pill with 372 milligrams of lactate. And every 50 pounds of body weight got one more pill. So if you weighed 150 pounds, you would have got, and they rounded up to the nearest 50 pounds, or not even the nearest, but to the next 50 pounds. I don't see what the point of measuring this against the placebo is. Like, we all know that adding calories before a workout is going to increase performance, usually. And so I think it would have been much more sensible to compare this against isocaloric glucose instead of a blank placebo. They don't clarify whether it was isocalorically controlled, and I think that makes a difference in interpreting whether this 4% increase in work is a reflection of the uniqueness of the lactate supplement versus the caloric supply. If you look at the discussion, they overview the other lactate supplement data. So time to exhaustion during constant load was unaffected by the addition of lactate to a carbohydrate sports drink. Bicarbonate and pH were greater during 3 hours of constant load cycling following ingestion of a polylactate solution, but no mention of a exercise performance benefit. Time to exhaustion during short duration high intensity treadmill exercise was barely extended less than 2% by high doses of lactate ingestion. Neither 20 nor 40 kilometer time trial performance was influenced by lactate ingestion. Okay, so far, every performance-related metric is negative in all of these studies. Time to exhaustion during high-intensity cycle ergometer exercise was appreciably extended 17% by high doses of oral lactate. So that's the one outlier. More recently, other commercially available supplements containing lactate have been shown to have unappreciable effects on skeletal muscle endurance during resistance exercise, but interpretation pertaining to lactate supplementation per se was complicated by the addition of other potentially active ingredients. All right, so as it stands, there's quite a number of studies, all negative regarding actual performance, one showing a pH effect. But this 17% increase in high-intensity cycle ergometer exercise time to exhaustion is the one outlier. So if we look at that study, the reason I struggle to take this seriously is the lactate is 20, 120, or 220 milligrams of lactate per kilogram of body mass in the form of calcium lactate we're looking at like on the order of like 15 grams of lactate, and that's a considerable amount of energy being provided to compare it to blank placebo, right? The placebo is aspartame and water, right? So you're definitely getting, a potentially significant caloric source. And I just don't think that's the right control to try to ferret out whether there's something unique about lactate and giving you a 17% extra time to exhaustion. So I honestly don't take these trials all that seriously unless they're isocaloric. And they're overwhelmingly negative anyway. So the way that I view that is as follows. Everything that we said about lactate so far means that lactate does what it does best when your cells make it from pyruvate. If you make lactate from pyruvate, it generates NAD+. If you make lactate from pyruvate, it alkalinizes you. Lactate, supplied exogenously, is not going to alkalinize you. It really can't because there could be some very indirect effects, but the pKa of lactate is down in the mid 3s, which means that it's not going to absorb a proton unless you're 1,000 times more acidic than you'd ever get in a maximally exercised muscle cell. And if you oxidize lactate into pyruvate in order to burn it for energy, you are generating NADH in the process, and you are releasing a proton in the process, which is going to be acidifying. It's not going to generate NAD+, and it's not going to generate an alkalinizing effect. So I just don't see how it is going to provide the same benefit that you would get when you make lactate yourself. So I think better strategies would be ones that we didn't really cover here. For example, boosting intracellular carnosine levels with beta-alanine supplementation can increase your muscle-specific buffering capacity. Acutely, bicarbonate, glutamine, and organic acids such as citrate, alpha-ketoglutarate, malate, fumarate, succinate can all be bicarbonate sparing because they are anaplerotic and they fill the citric acid cycle without needing the pyruvate carboxylase reaction, which sucks bicarbonate out of the system. And so all of those can be alkalinizing. Bicarbonate, you provide the bicarbonate, everything else spares bicarbonate in that list, and that will increase your acute buffering capacity. And it makes a lot of sense to also train for increasing your endogenous productions against buffering acidity, such as if you have, and I'm not a trainer and I'm not an exercise physiologist, but just reasoning through this, if you had a performance event 3 months from now and it's, you're off season now, or 6 months from now, let's say you have a performance event that's 6 months from now, you might want to spend 3 to 4 months training without the crutches to increase your endogenous buffering capacity. And then you might want to load beta-alanine to improve your carnosine stores as you are going a couple of months into the performance event. And then you might want to supplement strategically around specific training sessions in a periodized manner or the actual performance event to acutely increase your buffering capacity even beyond what you've trained endogenously when the time requires it. And I do think it would be very interesting to study lactate more. I just don't think that lactate supplements make the most sense in the context of viewing the alkalinizing effects of lactate. It's producing lactate endogenously that is alkalinizing. It's transporting it out of the muscle cell, not into the muscle cell, that's alkalinizing. And so while lactate is an excellent fuel source and not a metabolic waste product, I think most of the benefits you get from lactate come from when you make it out of pyruvate. All right, this has been Masterclass with Master John, Energy Metabolism lesson on how lactate alkalinizes your muscles. If you are not taking the full course and you've only seen this video, check out the link in the description to the full course. And I hope to see you in the next class.